Hi everyone. In this video, we are going to see about the genomic organization of eukaryotic cells. Genome is thus the entire collection of genes and all other functional and non-functional DNA sequences in an organism in a haploid set of chromosomes. It includes the structural genes, regulatory genes, and non-functional nucleotide sequences. Structural genes, the DNA segments that code for the specific RNAs or proteins. Functional sequences, regulatory sequences that occur as regulatory elements, initiation sites, promoter sites, and operator sites, etc. Non-functional sequences, introns and repetitive sequences. They are needed for coding, regulation, and replication of DNA. Much more in no functional sequences. Genome of all living organisms represents their hereditary material and is formed of DNA. In prokaryotic cells, a genomic DNA forms a singular circular chromosome without basic proteins that lies in the cell cytoplasm in nucleoid region. In an eukaryotic cells, DNA is associated with basic proteins, that is histones, in the form of long chromatin fibers. Chromatin fibers form a network that is enclosed in a double-layered nuclear envelope which condenses into chromosomes during cell division. Eukaryotic genome. The nucleus is heart of the cell, which serves as the main distinguishing feature of the eukaryotic cells. It is an organelle submerged in its sea of the turbulent cytoplasm, which has the genetic information encoding the past history and future prospects of the cell. Nucleus contains many thread-like coiled structures which remain suspended in the nucleoplasm, which are known as chromatin substances. Chromatin is the complex combination of DNA and proteins that makes up the chromosomes. The major proteins involved in chromatin are histone proteins, although other chromosomal proteins have a prominent roles also. The functions of chromatin is to package DNA into smaller volume to fit into the cell, to strengthen the DNA and to allow the mitosis and meiosis, and to serve as a mechanism to control gene expression and DNA replication. Organization of chromatin. In resting, non-dividing eukaryotic cells, the genome is in the form of nucleoprotein complex, the chromatin. The information stored in DNA is organized, replicated, and read with the help of a variety of DNA binding proteins. Structural proteins, histones. The main structural protein found in the eukaryotic cell is the histones. Low molecular weight basic proteins with high proportion of positively charged amino acids. Histones are bounded to DNA along most of its length. The positive charge helps histones to bind to DNA and play a crucial role in packing of long DNA molecules. Functional proteins, that is, non-histones, they are associated with gene regulation and other functions of chromatin. Nucleosomes are condensed several times to form the intact chromatids. Chemical composition of chromatin. DNA 20 to 40%, they are the most important chemical constituent of chromatin. RNA 5 to 10%, they are associated with chromatin as ribosomal RNA, messenger RNA, and transfer RNA. Proteins 55 to 60%, they are associated with chromatin as histones, a very basic protein constituent about 60% of total protein, and almost 1:1 ratio with DNA. The five types of histones are H1, H2A, H2B, H3, and H4. Non-histones, they are 20% of total chromatin protein. Then 50% structural actin and L and B tubulin and myosin functions during chromosomes condensation and in the movement of chromosomes during mitosis and meiosis. 50% include all enzymes and cofactors involved in replication, transcription, and regulation of transcription. Ultrastructure and organization. It includes multi-strand model, folder fiber model, and nucleosome model. Nucleosome model is the common one. DNA is tightly bound to histone proteins which serve to form a repeating array of DNA protein particles called nucleosomes. The three levels of organization includes DNA wrapping around nucleosomes, the string on beads structure. A 30 nm condensed chromatin fiber consisting of nucleosome arrays in their most compact form, that is solenoid structure. Higher levels of packing into metaphase chromosome. It includes the loops, domains, and scaffold structure. Euchromatin. Types of chromatin. Euchromatin and heterochromatin are the types of chromatins. Euchromatin, it is a lightly packed form of chromatin that is rich in gene concentration. It takes up light strain and represents most of the chromatin that disperse after mitosis has completed. It consists of structural genes which replicate and transcribe during G1 and S phase of the interphase. It is considered genetically active chromatin since it has a role in the phenotypic expression of the genes. DNA is found packed in 3 to 8 mm fiber. During metaphase, euchromatin takes up the dark strain. Heterochromatin. It is a tightly packed form of chromatin. It takes up the deep strain during interphase and prophase, but metaphase takes up the light strain. Chromosomes, centromeric regions, and knobs also take up the dark staining, of which centromeric regions and knobs are the true heterochromatic. Chromosomes are transcribed, so not true heterochromatins. In the chromosomes, all the centromeres fuse to form a long heterochromatic mass called chromocenter. Heterochromatin consists of a highly repetitive DNA sequences. It is late replicating during the S phase of the cell and is not transcribed. Euchromatin is uncoiled, active, and whereas heterochromatin remains condensed and inactive. Euchromatic chromosomes demonstrate complex organization characterized by repetitive DNA. Repetitive DNA sequences are repeated many times within the eukaryotic chromosomes. This table shows the classification of repetitive DNA. The satellite DNA is highly repetitive and consists of short repeated sequences. Centromeres are the primary constructions along the eukaryotic chromosomes that mediate the chromosomal migration during mitosis and meiosis. Telomeric DNA sequences consist of short tandem repeats that contribute to the stability and integrity of the chromosomes. Moderately repetitive DNA includes variable number of tandem repeats, mini satellites, and microsatellites. Short interspersed elements, that is, SINES, SINES, and long interspersed elements, lines, are dispersed throughout the genome rather than tandemly repeated and constitute over one third of the human genome. These transposable elements are generated via an RNA intermediate and are referred to as retrotransposons. The vast majority of an eukaryotic genome does not encode the functional genes. Only a small portion of the eukaryotic genome constitute a protein encoding genes. There are also a large number of single copy of non-coding regions, some of which are called as pseudogenes. Thank you.